PhD from Banaras Hindu University, and afterwards she has postdoc fellowship at CIML France, followed by Colorado State University in US, and then National Institute of Health, NIH, USA. And later on she had been professor at BITS Pilani, and then after that she is at CBLU. So she is working on the mosquito immune system and interaction with the Plasmodium as well as dengue virus. So I would like to invite Professor Lalita.
Thank you, Professor Singh and Professor Sanjeev Kumar. It is my great pleasure to be present over there. I would like to thank the organizer, Dr. Alok Bharti, for giving me this opportunity to present my research work. Here I am switching from COVID-19, which is an excellent work done by Dr. Bhatt, who was here, to mosquito immunity. So it's like a kind of switch, and I hope you are able to understand that though we recently gone through this COVID infection, mosquitoes and the diseases transmitted by mosquitoes are also very, very important. During that time, I remember, like when COVID is going on, so many dengue infection is going on. It's very difficult to find it out. It's really a dengue infection or it's like a COVID infection. Everybody was like kind of scared. So today, what I'm going to present over here is about the transcriptome analysis of ABC transporters in immunity against blood-borne pathogens. So before I would like to give some introduction and why we are working over here, what is ABC transporter and what we know about mosquito and its immunity. So just now we have learned about like we humans are vulnerable of different diseases. Here we talk about direct infection, which is like a COVID. We just learn about how this COVID is affecting us. Here are some blood-borne pathogens where we talk about indirect. Here we require two hosts. So when we talk about two hosts, like here, in case of blood-borne pathogens, we are talking about the one host is mosquito and the other host is the human. So I'm giving you two pictures just as a glimpse. Like here is a malaria, which is transmitted by Anopheles stephensi, and there are several species of Anopheles. And now the other side is the dengue, which is transmitted by Aedes and many species of Aedes. So there is always easy to target the human host. But when we talk about the mosquito host, it is important because if you think about, like here I'm giving just examples of several other species which transmitted disease when even killing the people. Mosquitoes is one of them. And because they are able to transmit dengue, malaria, West Nile, chikungunya, and variety of bacterial and viral infection and plasmodium infection. So I would like to go why we are why we are looking for mosquitoes. Because when they transmit several diseases, there are different ways to control it. One way is control is just for protection, as we all know that we can get the vaccinations. On the other ways, like we have got different anti-malarial drugs and other drugs which we can protect ourselves. On the same time, we just looking at the transmission blocking. So if we able to block the transmission from mosquito host to the human host, that will be easy. So we can not allow parasite to go inside the mosquito. In that case, this term we use as a transmission blocking. One way we can just even kill the mosquito through the insecticides. On the other way, we can prevent ourselves, just we are using different like bed nets we are using repellent. And one of the very important and emerging one is like a blocking parasite development inside the vector. And when we say inside the vector, we have to know more about vectors. So I'm just giving you first two glimpses of one. I worked on like a little bit on killing the mosquito because this is easy, we able to do experiment very easily. And then we move towards the blocking the parasite development. So you can think about the mosquito immunity and how they able to control parasite development inside the mosquito. So just giving a one insecticide example. In case of that, we were looking for when we came from US and with Filani, like we were just like setting off mosquito lab is not very easy. So we were setting the mosquito labs, same time the parasite and everything, and at the same time we were saying, okay, let's look for any biological way which are environmental friendly, we can control the mosquito population. So here I am giving the one example of agave augustifolia. This plant is present in the Rajasthan area and towards the desert, and very specific that when we take just water extract, they have a efficacy to kill the mosquito larva up to 100% within 48 hours. So, and important thing is that they give the activity only in time of summer. As soon the winter comes, no activity. So we were surprised that the student might not be doing correct experiments. All of a sudden it's required that we thought, okay, let's change the temperature. So when we change the temperature 37 degrees, again this plant extract is giving the results. So we published over there in the journal Frontiers, and now even government is also wants to know about this plant, and they are looking more how we can find it on different extracts and use it for just killing mosquitoes. From here I am just switching towards the mosquito immunity. So once we know how the mosquito immunity is able to kill the parasite, we can easily block the transmission. So mosquito have a very strong immunity. One is called pattern recognition receptor, so its innate immunity is present. And they also have a signaling molecules like immune signaling pathway. I am working on JAK-STAT pathway. So JAK-STAT pathway is very well characterized now in mosquito, and we found that this JAK-STAT pathway is actually working as a negative, positive regulator, is actually suppress the plasmodium development. So just for students who are not in this field, this plasmodium, they enter into the mosquito and it almost takes 21 days to stay inside the mosquito, and the mosquito is able to transmit the disease. And we are looking at the stage, because this is the midgut epithelial, and we are looking in the stage when this becomes a oocyst. So it's better to, if it is possible, we can control this transmission because it is a midgut epithelial, and if we control over here, that is much easier. So I'm just giving an example of STAT signaling pathway. We silence the STAT molecules, and when we silence the STAT molecule, we find it out in case of Burgeri, we find that this is increasing the parasite load. That means we calculate on each individual mosquitoes the number of oocysts present over here. On the other side, we find it out that in day 7, when we talk about the day 7, it's like of SOX gene, which is the suppressor of STAT pathway. This is going down. That means it's suggested that STAT pathway is activated, and when we silence, that the parasite number is going up, and when we have a SOX, then the number is going down. And this interesting finding, we found out that not only the early stage of oocyst, but even the late stage of oocyst, this STAT signaling pathway is there. So from here we are started looking all more about the STATs, and here we find it out like there are several STAT molecules are present in 18 Anopheles mosquitoes. Some having a STAT A as a conserved one, which have a different introns, whereas a retrotransmission there is a STAT B. And actually STAT B is regulating the STAT A. But in few mosquito species like Anopheles sinensis, Dyrens, Dylangi, Albinus and Ferropi, they have a single STAT. So when we talk about any like control mechanism, and we say it's easy, now we find it out molecules and it can be easily controlled. We find out there are so many types of plasmodium present, and there are different in mosquitoes. So combination of all these to have a mosquito and parasite and finding out one target molecule, because still is going on, still we are looking for. After that, we move to one of the molecule which is present inside the mosquito, and that is also an immune molecule, that is apolipoforin, and here we also did the silencing experiment. It's a big experiment, it takes like almost 12 days, because we have to use the double-stranded RNA, inject inside the mosquito, gave the plasmodium infection and allowed the mosquito to survive, and then started counting the parasite. So I am giving here the example of apolipoforin 3. This is known for flight lipid proteins, but they are also play a very important role in mosquito. So here when we see that laxity and when there is a apolipoforin 3, the infection is going down. So that is actually working as a negative regulator. So when there is a no apolipoforin, parasite number is going to be increased. So it's actually helping parasite to grow. So here I'm just saying that we are working hard to find out few molecules which going to target towards the plasmodium development inside the mosquito bite. And in that case, we are finding new and new molecules. Now I'm coming from here towards the ABC transporter, which has been recently started working. And these ABC transporter, why started working? Because we find out some ATP transporters, they are actually having a role in insect immunity. Some ABC transporter immunity, these are the some papers which coming out. And from here we also search the literature, and this we find out that some like in case of wheat or in case of other molecules, they are actually transporters are sensitive to or suppress the pathogen development. And single mutation in wombat we find out that with the ABC transporter resistance to the BT toxin. And there are a few more examples where we say yes, these ABC transporters are working. I'm just giving, I think enough 2 minutes. Okay. So here we would like to know how this ABC transporter, which is ATP binding cassette transporter, and whether it's working in mosquito immunity. So this project was funded by DBT, and we are looking for some novel immune molecules which may regulate the pathogen development and might offer a new insight to interpret the mosquito cycle of bloodborne pathogens. I'm giving this example how this ABC transporter looks like, and in Aedes aegypti. So ABC transporter have been 2 domain. One is called nucleotide binding domain, and other one is transmembrane domains. This nucleotide binding domain is actually having a ATP binding, like you can see over here, and they have a transmembrane domains. So transmembrane protein that transport amino acid, sugar, peptide, small lipid across the biological membrane. And as I said, they have a NBD which having a Walker A signature sequence, Walker B motifs, and they have a signature sequence. And TMD, they have a 6 helical embedded in the bilayer membrane. So this is a ABC transporter proteins. Based on how many ATP binding cassettes they have, there are 2. One is called half transporters, or you can say full transporters. You can see this is the half transporter and the full transporter. This is the mechanism how it working. That molecule is coming, ATP is and able to release the molecule over there, and it's released ATP. So what we are looking over here, so I am talking about Aedes aegypti mosquito, and in Aedes aegypti mosquito we are first identifying and characterizing the ABC transporters, and then we are looking for their expression in the mosquito development and identifying their expressions and find with the immune challenge. We are giving the immune challenge of bacteria over here and finding out if there is any molecules. So reverse genetic approach we are using to find out the specific molecule of ABC transporter. So with the literature search and the database present over here, we are looking for ABC transporter, and in case of you look over here in Aedes aegypti, there are almost 59 ABC transporters are present, and they are having a subfamily from A to H. We compare like in Drosophila, Anopheles, Bombax, Culex, as well as Homo sapiens. What we find out that ABC A, C, and D subfamily are widely expanded. So we published this paper last year in Pathogen, and afterwards we are also starting looking like whether this ABC transporters expressing inside the mosquito gut or in the carcass. So here are the some genes which we say, like in case of ABC, Some are expressing both in midgut as well as in carcass, whereas some are only expressed in carcass or some are only expressed in the midgut. So there are some very unique because they have a different domain structures over here. So we started targeting few of them. How we targeted? We are looking for some molecules, like here I am giving the example of ABC B2, C10, G1 and G3. We did the experiment giving the live bacteria challenge with these mosquitoes as well as heat-killed bacteria challenge. First, over here you can see the different time kinetics from 1 hour to 12 hours and looking the defensive, which is very known molecule, antibacterial molecules, and this expression is going high shifted the related mRNA expression. B2 of ABC transporter B2, this expression is there but not very high. Same way in case of C10 and D1 and G3. So this expression with this one, we can say that expressions goes up and down in some cases, and here in some cases you can say in C10 or G3 the expression is like going up and then again going up in 6 hours. So from there we are looking for different, this is a little bit very big slide because we are looking different ABC transporters and how these different families are working. So I am targeting only 1, 2, 3 over here, and this is the 4th one and 5th and 6th. When we challenge, so we say ABC transporter of subfamily A, C and G are induced after both the bacterial challenge, if it is heat kill or if it is a live. Then we looking for one important one that is called ABC G3 because they have a similar to E. coli toxin antitoxin system, and we find out that how they are similar to other and where it is found. So we use KEGG's sequence similarity database to analyze this ABC transporter G3. Now from here we plan to silence this ABC G3 genes through RNAi, analyze its effect on blood-borne pathogens. So here you will see that there is PCR amplification of ABC's G3 genes of 4 sets of midguts we are using over here, and then we clone it and make a double-stranded RNA of them, and finally you can see the double-stranded RNA of ABC's G3. And after that we did the silencing. So here is the silencing results. We make a double-stranded RNA of G3. This is the control. There is a 80 to 90% reduction in the mRNA level, and then we are looking for their bacterial load. So we find it out that when the gene is silenced, the bacterial load is increasing. In case of sugar fat, as well as when we give the bacteria supplements, also it is increasing. We suggested that ABC G3 is actually working against this mosquito, Aedes aegypti, and is working as an immune molecule. We look for another gene, that is ABC C12, and we did this experiment to find out their viral load when there is a dengue infection. And from here, this experiment is preliminary experiment. We are also again looking into more details. So based on all these experiments, we are just giving that mosquito having a strong immunity, they are able to kill the parasite inside, but we have to find it out exact molecules or the different targeted molecules over there. And when we talk about new molecule ABC transporters, we find it out there are 59 ABC transporters are present in Aedes aegypti, and they have a gene or gene segments duplication. They are widely expansion of A, C and G subfamily, reveal their importance in the mosquito biology, and suppressor of immune ABC transporter is the survival strategies of the virus. There are different scopes. We can also work again currently the antigen specificity of innate immunity, suggesting to explore the list of transporters and other immune genes in the regulation of virus development, and designing the strategies to manipulate this important molecule for blocking the pathogens development and transmission. So I would like to stop over here, and I would like to acknowledge The stat work is done by IL28 Gupta, Apple is by Rini. Professor Sanjeev is with us, doing all sorts of experiments and helping the students. And whenever we stuck, this is my team. Here's the Niklesh who is working on this work, and this is students that I am doing is working on daily. Thank you so much.
On the halfway signs, please raise your hands. Yes.
Have you tried silencing the transporter?
No. It's just like first of all, like you have to target one organ. So we target the gut, salivary gland we haven't tested.
The other thing, so initially I think the bacterial infections you mentioned, you were using a mixture of live and dead bacteria.
Yes.
Is there any specific reason?
No, actually it's not very specific reason. We just would like to say as a antigen elicitors, how they are working. So if it is a heat field, that means it's going to produce molecules. When we are using live, what there is an effect. That is the one.
Any?
So Lalita,
Yes.
good lecture, good piece of work. Just a couple of observations or questions, whatever you can say. You have done work on ABC transport,
Yes.
right? So is it possible at this juncture, can you correlate the dynamics of this ACP transporter genes with the propagation or development of this pathogen, for example in malaria, mosquitoes we were talking, or you can say their transmission, I mean can it be correlated with the development of the pathogen and its transmission, transporter genes and the transmission?
Okay, sir. In that case, like we started with the just first to align all these ABC transporters present inside the mosquitoes. And first we find out like they are in, there are so many duplications over there, and almost they have like one say ABC A, there are also several ABC A transporters. Some of them we are targeted, like we just talk about G3. We give the bacterial infection, and yes, there it's showing, when we silence is showing, yes, it is working. But further we have to do more experiment to find it out how they are controlling the antigen or any virus infection.
Okay, just give me one or two ideas.
Yes, sir.
In Singapore, there was a dengue problem.
Yes,
There is one company, Oxitec, they released transgenic insects. So I think if you can catch hold of those people, I mean you can see the transporter gene mechanism in those mosquitoes
Yes.
which have been released for that purpose.
Sure.
Basically, the concept is based on sterile insect technique.
Yes.
And for the last 3 decades I have been working on that. So my interest is, if you can correlate that, then I would come to the next question whether the radiated insects can have some sort of mechanism working over there or not. And another point is that suppose you are able to find out this innate immunity, will it be possible that you can use some efficient method to block the transmission?
Yes, sir. In case of like Plasmodium, when we talk about Plasmodium, they are looking for the molecules, and especially when we are the salivary glands. So in the salivary glands, they are finding out the sporozoites during that time and how they can able to block the transmission. There are several molecules which are working over it.
Thank you. I don't know whether you are remembering or not.
Yes, sir.
This couple got the professor's position. I was incidentally the expert in that committee. So,
Thank you, sir.
Please. Any of the STAT inhibitors can also be a very good molecule to inhibit the virus. What sir is asking to block the...
Sir, actually, when we look for the starts in the pathway, we find out the NOS. And NOS is really this very well known, and like even Sanjeev, Professor Sanjeev is also working on virus series and NOS. And that is, we are planning to really look for that as a
plant extract you are showing that has some ovicidal property also. It can kill at the egg stage.
At this stage, we
That will
haven't
be more
checked it.
advantageous, right?
Yes, we haven't checked it. But of course, it has a mosquito stage, but not egg stage. We have never checked it. No?
Check on to the larval
Larval
stage.
stage.
And it can
it has ovicidal property, it
Yes.
will be all the more good.
Now they have seen water extract, they may see different types of extracts, you know.
No, whatever the extract we tried,
Both. This
but
is
we
the-
only stick to the water one because preparation is easy and we are avoiding the chemicals. So water one must be least.
No, water extract is fine, but,
But he's looking
instead
for
of
egg.
larvicidal, whether it has ovicidal property, means it can hatching up egg can be avoided, no?
Yes,
we
he
never
wants.
try it.
We
We have to
can
try that.
try that.
That is more advantageous, more preferred one.
I think there are a couple of things very interesting one can do. I can give one idea at this moment.
Sure.
I may not forget that. There are certain photosensitizers and they are very well documented, but very less work has been done in that case. So photosensitizers are very safe. At the same time, India has got no dearth of sun. The moment you use those chemicals and there is a sunlight, they will immediately, they will be toxic. Alpha T has been, it has been proven a very good insecticide against these mosquitoes as well as lepidopteran insects. And we have been giving the data about bombics, and these are only two groups, lepidopteran and different. Photosensitizers का थोड़ा सा देखिए, उसको इसके साथ जोड़ेंगे, it will be great.
Thank you. Thank you.
Thank you, Professor.
I request Professor R.K. Seth to please felicitate Professor Lalita Gupta for delivering such an exciting